Introduce yourself.
Hi, my name is Mark. I'm doctoral candidate 7 within the ORGESTRA consortium. I work mainly here at the University of Lisbon within the Creative Sciences Institute, but during the course of my joint PhD, I'll also be working at KU Leuven. I'm originally from Ireland, where I did my bachelor's degree in medical science and a master's in biochemistry and pharmacology.
What made you want to work in science?
Well, I've always been interested in how things work, including human biology, and particularly during my master's and my bachelor's degree, I became quite interested in RNA-based therapies.
So what are you up to today?
So after we filmed the video this morning, I have some experiments planned treating human bronchial cells with new therapeutic compounds that we designed.
What do you like best about your workplace?
So I'm working here in the cystic fibrosis lab. We're all working on the one genetic disease, but from different aspects, so I get to get a lot of different feedback from people on my project.
Tell us a bit about your project.
So I'm developing a gene-based therapy for cystic fibrosis. This is a genetic disease that causes thick, sticky mucus to build up in the lungs and other major organs, and that leads to difficulties in breathing and other digestive issues. The therapeutic molecules I'm developing are called antisense oligonucleotides. In our cells, information in our genes is first transcribed into messenger RNA and then translated into a protein. People with CF have mutations in their DNA that causes a change in the mRNA and in turn leads to a less functional or completely absent protein. Antisense oligonucleotides work by binding to the region in or around the mutation in the mRNA and helping the cell to produce a correct version of the protein even if the gene itself is mutated. In CF, this correction helps the protein to reach the cell surface and transport chloride ions properly. This allows the body to produce normal hydrated mucus and reduces symptoms significantly.
What is the impact of your research on society?
Well, around 20% of people with CF aren't eligible for any treatment regimen, which means they're not able to benefit from the current treatment regime, which is CFTR modulator therapies. We aim to use cells that we obtain from patients to develop three-dimensional mini organs that we call organoids. These are advanced models that still carry the same mutation as the patient, and we can deliver our therapies into the organoids to predict whether or not a patient will respond to a drug. The personalized organoid testing protocol that we developed should allow for the rapid and relatively easy development of antisense oligonucleotides that we could offer as new treatment strategies for the patients who have no eligibility for other treatments.
If you were in an orchestra, what instrument would you be?
I guess I'd probably be a violin, a bit high maintenance, but quite versatile and precise when it's in the zone.